to frame this a little bit in exploring the Gene Curation Coalition database for biocurators. So the live demo that I'll give will be a little bit different from just the consortium call, just to tell you how you could use it if you're a curator curating gene-disease relationships, for example. And we'll also talk about some of our new work. We actually just have a new set of submissions that we processed that aren't quite live yet, but really do increase the numbers of curations in the database and some of our conflict resolution. So I'm excited to talk to you about that. So just an overview. First, I'll tell you about the GenCC and why we thought we needed it and thus formed it. And then the Delphi survey that we performed a couple of years ago now to harmonize clinical validity terms across all the GenCC member organizations. And then I'll end with a live demo of the GenCC database and tips and tricks to use it for ClinGen curation. So what is the Gene Curation Coalition? I don't think I need to tell any of you on this call, but it's really critical to have curated gene disease databases. And this really can be visualized when you plot just, for example, tests for hypertrophic cardiomyopathy from 14 different US clinical labs with the number of genes, and see that they vary really widely. So we felt informing this GenCC that really standards and consensus are needed for which genes are valid disease genes that are ready for clinical testing. And of course, you all know that being ClinGen curators. And then another level of that is different levels of evidence are needed for different clinical uses, perhaps higher levels of evidence for population screening and predictive tests versus diagnostic panels where prior probability of disease is already much higher. So these are just the GenCC members, and I am really happy to report that with the launch of the database, our membership is growing monthly, really. So we have public gene level resources, and of course, ClinGen is one of those, and a many handful of others internationally. And then I'm also very excited to report that we have some private gene level resources. And so a lot of these gene curations that have been done by these clinical labs actually hadn't been previously available. So they very graciously agreed to share their data on this open source database just for everyone's use to really help facilitate gene disease curations and patient care in turn. So we had a few goals when we started the GenCC. First, when we started, we really wanted to understand the approaches and classification systems of the different curation efforts, because different efforts really did have different purposes. And we didn't want to necessarily be comparing apples to carrots. We wanted to all be on the same page. We then wanted to develop consistent terminology for validity assessment, as well as inheritance, allelic requirement, and mechanism of disease. And I'll talk to you about the validity assessment terms, as I mentioned, but the terms for allelic requirement and mechanism of disease are well underway too. And we're working with the variant effect predictor, or VEP, to really integrate some of these in the sequence variant nomenclature and also copy number variant. So stay tuned on that. And then we wanted to post gene curations from any group willing to share with the public. So the database I'm going to show you really is like a ClinVar, but for genes. And we then wanted to use this database to resolve differences and collaborate on gene curation projects. And I'll speak a little bit to that at the end. So first, when we were looking at curation efforts across the different groups, we found that they really do differ in gene curation results. And one of the first efforts that we looked at was ClinGen versus an effort from TGMI previously that's been discontinued, at least for now, which is the gene disease map. And so, one reason that maybe these differences may arise is the granularity of the curation. In this case, the gene disease map was really just a very broad yes or no is a gene potentially associated with disease. And as you're all quite familiar with, the ClinGen gene disease validity framework is very granular. And if a gene has multiple diseases, it may have multiple curations. So already you can see why there would be some differences across these curation efforts. So when we compared, red would be yes, are mutations posed to cause disease, and greens would be, of course, definitive for us. There are really some discordances. So we have the limited, refuted, disputed for ClinGen, and then some genes that are not posed to cause disease or posed to cause disease. So we really do have discrepancies across in this effort and comparisons here. pointing for standardization. And another one of the groups within the GenCC, the PanelApps, both Australia and UK are actually on the same system, facilitating the comparisons between the two groups. And so they compared about 2,700 genes and reviewed 1,000 or so discrepancies, and really helped to resolve some of those discrepancies across these different panels. And one other activity that we did to really start to understand how we all curate genes is we actually took a handful of genes and classified them across all of the different groups. And you can see here that these are color coded. So green ones are really meaning more towards the moderate, strong, definitive area, like these are causing disease, whereas yellows are maybe uncertain, limited, moderate, and red's really not causing disease. So you can see that for a particular gene, they're all across the board as far as how we curate genes. So we really felt like we had to understand why and how everyone was curating, and also work to resolve some of these discrepancies. So during these pilots that I kind of mentioned for those three particular examples, it really did identify some reasons why there was discordance. So I spoke to this a few slides ago, but definition of disease, maybe there's high penetrance versus inclusion of lower penetrance phenotypes when you're thinking about disease and curating that claim. So that can be discordant. The purpose of the curation, the clinical validity framework, for example, for ClinGen versus whether you should include it on a panel based on a phenotype. Just because ClinGen has something as strong or definitive, another, for example, the PanelApps may not want to include it on a panel that's not necessarily associated with a particular phenotype. So they might have it as red, which would be seen as a discrepancy, but because of different purpose of curation reasons. Another one that could cause discordance are differences in disease and phenotype assignment. As I mentioned, genes often have claims for multiple diseases. So sometimes these are lumped, sometimes these are split. Sometimes the groups do different things to lump and split conditions. Or if it's very broad, like the gene disease map, like I mentioned, it might just be, is it associated with disease or not? Yes or no. And then, as we know, published literature can change pretty rapidly. So the date of evidence evaluation could really make a difference. And you've seen this in ClinVar too. If one group evaluates it 2 years ago and another group evaluates it today, it could have very different clinical validity classifications. So understanding the differences and focusing on resolving them did really require harmonization of terms and definitions. And it started out pretty confusingly. So these are kind of all the terms that are used across some of the original resources. And for an example, so we use words. Some groups use colors, but actually opposite colors. So red meant associated with disease in the GDM map that I talked about, whereas for PanelApp, it meant not associated with disease. So this was really very confusing. And if we were to place all of these on a website, the community would just be very confused. So we really felt like we needed harmonization across all of these colors and terms and present or yes, no, which is why we embarked upon the Delphi survey that I'll talk about. So in order to perform this Delphi survey, we all had to be on the same page. So we drafted harmonized definitions for the gene curation categories. And then in the framework of a Delphi survey, we had multiple rounds narrowing down terms through each round. And the first round, we solicited term suggestions from the GenCC members. So for round one, we just surveyed the GenCC members, which was 33 individuals. And then in the second round, after narrowing down some terms that weren't voted for or had low scores, we extended this to the membership of the GenCC group. So some of the curators, for example. And then in the third round, we surveyed the international genetics community and included a paired optional explanatory video that was about 7 minutes or so that really explained the purpose and a lot of the slides actually that I just showed you as to why it's really critical to harmonize gene curation efforts. And we were lucky, we did get 241 responses, and I'll show some demographics about those responses now. So we asked about location, and most of the respondents, as you can see, were in North South America, this really just being the US and Canada more so. And then the other half are approximately were from the UK and Europe. We asked if anyone was from particular societies. And here are the surveyed populations that we reached out to. Of course, it's easiest to be able to reach out on society email listserv. So many people were part of these different societies, ESHG having the highest respondents and ClinGen actually coming in third. We asked people if they participated in consortiums, and you could have multiple responses in this case. ClinGen was the most participated in consortium, but we're also happy to see that many people did not participate in a consortium. We felt like this was a good thing because we felt it was important to remove the bias of being very close to a consortium and its particular choices and names for gene disease validity. So we were glad to see that some people were at least removed from this consortium discussion. We wanted to ask people how familiar they were with genetics on a scale of 1 to 5 in case we had to stratify some of the answers based on genetic familiarity. Luckily, we didn't really have to do that. Most people were quite familiar with genetics. We did ask people how they would describe themselves, and multiple responses were allowed. The top answers being researchers, clinical genetics laboratory directors or staff, medical genetics.
Marina, sorry, I think your screen's frozen. The slides haven't moved on. Sorry to interrupt.
Oh, it's okay. Where did it get stuck?
I'm on where I can see comparison of terms. Sorry to interrupt you.
Oh no, please do. I am talking about nothing if I can't show it. Let's try again. Okay.
Thanks.
Is that better? Is it moving now?
I'm seeing a black screen at the moment.
Yeah.
Hmm. Can you see anything now?
Yeah, I can see GenCC term Delphi Survey process.
Okay, great. I will head back to demographics and present again. Thank you for interrupting, and don't want you all to have to look at the same thing over and over again. Okay, and you can see it now. Looks okay?
Yep, looks good.
Okay, great. So we did ask people how they would describe themselves, and multiple responses were allowed. Equal responses of researchers, clinical genetics laboratory directors or staff, medical genetics physicians, and so on. So after these rounds of Delphi surveys, here are the finalized terms that we actually use on the website: definitive, strong, moderate, limited, animal model only, no known disease relationship, disputed evidence, and refuted evidence. We were happy to see that round 3, which was surveying the community, had 93% term agreement. So 13 out of 14 of the terms, with round 2, which was just surveying the extended GenCC membership, really suggesting that the genetics community is harmonious with the GenCC. So as I mentioned, all groups mapped these terms on the GenCC website or supportive in the case of OMIM or Orphanet, although none of those are live yet, if not enough granularity to do so. So now I'll talk to you about the GenCC database, and I have to have a huge shout out to Scott Geringer, who's also responsible for a large amount of content on the ClinGen website, hence the streamlined beauty of the GenCC website, much like the ClinGen website. He's really worked so wonderfully with me to help get this launched. So just an overview of the submission pilot that many of the GenCC member groups had. So at least for now, we're using a spreadsheet-based submission. We're really hoping to have an API-based submission in the future, particularly for OMIM, because they really would like to ping us as often as possible. And we would like to have that for ClinGen too, because one caveat that I would mention is just because you search for ClinGen curations on the GenCC website does not mean that they are missing or that the gene hasn't been curated on the ClinGen website if it's absent from GenCC, just because as of right now, we have to do spreadsheet-based submissions. So there isn't live crosstalk between the ClinGen website and the GenCC website. So you don't publish something on the ClinGen website and it automatically goes over to GenCC. So that's one current caveat about our spreadsheet-based submission system. Curations are mapped to the harmonized disease terms that we generated by the Delphi survey, and as I mentioned, there's the addition of supportive for both OMIM and Orphanet. The disease terms, just to standardize them as much as possible for now, that we accept are OMIM, Orpha, and Mondo, and we perform harmonization with the monarch disease ontology. So if something, and the reason that we use this, as you know, is the Mondo ontology contains OMIM and Orpha IDs. So hopefully most of the time we're able to map these ones if they're submitted to a higher level or a synonymous term in the monarch disease ontology, which we're going to leverage for conflict calculation. I'll talk about that. And then a download is actually available of all the data sets, but OMIM. You have to go to OMIM's website to register for downloads, or if you're commercial, obtain a license for that. 8 groups completed pilots, and more of these groups are pending. And as I mentioned, we have had a few new members join the group too, so even more submissions, and hopefully more of this database will grow. And if you're interested in signing up for alerts for really big feature updates, or we're hoping to have monthly or quarterly updates as to the number of submissions, feel free to go to the GenCC website and type in your email. We won't overly spam you. So this is just a screenshot of the summary statistics that are live right now. We have almost 4,000 submitted classifications on about 2,300 unique genes from 8 submitters, most of those being definitive, and then strong and limited and so on. The latest update, I'm pleased to add, Scott really worked hard to get me these numbers. I'm still spot checking it with him, so it's not live on the website yet, but we're able to jump from 37 to 5,500 submitted classifications of 2,300 or so genes to 2,800 or so genes. And we have 1 increased submitter. And we actually just got a submission from OMIM that we're working on processing too. So we'll have a lot more than this, but I'm really excited that we have even this biggest increase from the last go live in December. So our next goal is to analyze the data to assess concordance and begin collaborations to resolve differences. We haven't yet analyzed the data, but we're starting to do that, particularly with conflict resolution. So we're hoping to get an overall analysis and describing the Delphi survey that I just mentioned out in a publication this spring. And so in the ClinGen community, of course, we trust you all, but we do have a Fort Lauderdale agreement on global analysis of this data for now. So in our ongoing work, we're identifying and resolving conflicts. So I want to talk to you a little bit about that. So we're trying to do this in phases because, as you know, the disease nomenclature, literature, and field is very discordant at this point, and it's decently obvious when we look at submissions in the GenCC, and it's making conflict calculation a little bit difficult. And that's why we need to do it in phases. So the easiest way to do it is exact disease term matches. So either, for example, groups are submitting the exact OMIM term together, or a monarch disease ontology term that is mapped to a synonymous OMIM term. And so if these have differing classifications, for example, a definitive one and a limited one, and they have the same mode of inheritance and the same disease term, we would calculate that as a conflict. Very simple. For phases 2 and 3, we're hoping to use broader disease terms for conflict calculation, and I'll speak a little bit more about that. Preliminarily, Scott was able to pull together a conflict report pretty rapidly, but there are preliminarily 90 conflicts of exact disease term mode of inheritance matches. So in this case, we're defining conflicts as those genes grouped together as definitive, strong, or moderate versus all of them that are below that, so limited and below. And at this point, there are out of 5,500 or so are about 90 conflicts of this type. And this is just a mock-up for phase 1 of being able to generate a conflict report. So in this case, we'll have editable conflict parameters where you can set certain of the groups into group A and certain of the other groups into group B. And if you wanted to exclude, for example, animal model only, you would be able to exclude that. And then you'll have this very nice page where your conflicts are highlighted and you can download a conflict report. And this is just a screenshot of the editable conflict parameters where you would have 2 groups that you would be comparing one versus the other. And you can include them all. If you wanted to include all of the clinical validity categories across the groups, you can do that, or you could exclude one if you felt that it wasn't relevant to necessarily calculate conflicts. Or if you wanted to see, for example, definitive versus limited, you could exclude everything else and just look at those two. So we're trying to make it as flexible as possible. And GenCC will have a recommended setting, so we'll also make that clear too. So for phase 2, we're hoping to have database level disease grouping choices. So editable conflict parameters where a user has a login and can save their searches, and a downloadable conflict report. We're hoping to, instead of using exact gene, disease, mode of inheritance matches, to be able to use broad ontology choices made across the entire data set. So something like treat all disease terms in an OMIM phenotypic series as disease matches. Because from what we've seen, oftentimes a group will submit something, for example, with the very specific gene level OMIM term. So deafness 37, for example, and other groups will submit on a higher level term, so just deafness. And really, in this analysis, we'd like to be able to count those as the same. The first phase, we wouldn't be able to do that because it's just a little bit higher of a computational ask, but we're hoping in this phase to be able to make broad choices like that as we continue to harmonize disease terms that we use. And then phase 3, which is the most complicated, is an ontology tree search and a panel builder. So we're hoping eventually to incorporate the monarch hierarchies within the database. And then the user will have the ability to calculate the conflicts based on their own rule choices. So disease keyword matches, setting all terms in a specific hierarchy as synonyms, just a lot more flexibility. And this will also have a login and the save function. And then hopefully the integration of Mondo will allow conflict reports and disease panel building.
Marina,
So for disease, yep, go ahead.
Sorry, can I just interrupt and ask a question? Sure.
Yep.
Actually, Deb had a question here in the chat. What do you use a conflict report for? And can you give some specific examples? The GenCC could use it to go back to groups to try to harmonize, but for the contributors, how would they use the conflict reports?
Yeah, that's a really good question. I feel like much like in ClinVar, how expert panels will use conflict lists, much like GenCC would use it. I do think that the submitters can also go back to their groups with these conflicts and start to evaluate how they choose disease names, start to evaluate how they classify their gene disease relationships. And a lot of this might just be differences in how they define the classifications of gene disease relationships. But I also can picture super users of this. For example, the Lab for Molecular Medicine recently reached out to me and they're trying to generate gene lists for their medical exome, for example. And they really wanted a broad list of all the genes with the particular phenotypes that were classified as definitive and were concordant. So I could picture some large gene list development for super users and that maybe those with conflicts deserve a second glance. Or maybe you need to do some manual analysis on your part to decide whether you include or exclude something based on that. The same could go for panel design, that if multiple groups are having discrepant classifications and you are a separate lab that's using this database and looking at it, that you might want to do some of your own analysis for that particular gene disease relationship to decide whether it's on your panel. Or if you're really interested in that, you could highlight it and contact the GenCC and say, do you think you could prioritize this gene? It's, for example, a gene therapy was just developed for it and it seems to be conflicting on the website. So those are some use cases that I could think of.
Great, thank you.
Mm-hmm. Any other questions before I go on to this future work? And then I'll do the database.
There's nothing else in the chat right now. Does anyone have any questions?
I do have, this is Deb, I have another one. So in terms of like the disease content, and if you, so I know that there are plans to split it based on disease content or disease groups. One area I'd be interested in, of course, is cancer and cancer genes. And so how do groups get involved with the GenCC if they want to contribute genes? Like I'd like to see NCCN genes curated, through cancer groups included too.
Sure, I can show you. So we actually have an FAQ on the website as to how to get involved and reach out to us. And we have like a general contact email that goes to, I think, me, Scott, and Heidi. So I can show you that FAQ when we get to that.
Great, thank you.
Definitely groups have reached out to us and said, this database is wonderful. I have a curated database myself of gene disease assertions. Can I please join your group? And we welcome people for sure. Great, so I will talk about just this last bit of future work, and then we can go to the website and I can show you around there. So this would be the most complicated of all, of course, is the ontology tree search and panel builder. So in theory, we would have a term search. You can log in so you can save everything, where users can search any term in the Mondo hierarchy or HPO phenotypes or disease name keywords. And then once you have whatever list that you've generated, you can filter this list by clinical validity or by submitters, anything that you would like, even mode of inheritance, for example, and then download the filtered list. And you could potentially use this for panel test design or indication-based analysis for exomes and genomes. And that is definitely the pie in the sky reach, but we're hoping to get there at some point. And just before I go to the database itself, I just wanted to acknowledge everyone in the steering committee. I know that a bunch of you are on this call and really do appreciate all the time that you've taken to assemble your submissions. I know a lot of that can be very manual, and just to contribute feedback to the website. And then of course, I really wanted to thank Scott and Phil to help with all of their work for the website and database design. Okay, so let's go to the website. So there are two ways to enter the website, actually. So the GenCC.org is largely the text-based website. So this, of course, links to the database, which I'll show you. But we have about, so it talks about all of the goals of the GenCC, basically why I talked to you why it was needed, the steering committee members. If you go to the members, you'll see all the different members with a little summary blurb from every single one and a link out to their website as to basically what they are and what they're about. And then some different news and articles. For example, we recently partnered with HGNC to stabilize the gene symbols that are within the GenCC database. And basically they're trying to prioritize any of the strong or definitive genes within the GenCC database and working on stabilizing those gene symbols. So we put an article here for the news updates. And then we have a few different abstracts that we've presented in the past. So you can either click on the GenCC database from here or this announcement, of course, or you could just go to search.thegencc.org. And no matter how you get there, it will take you to this Fort Lauderdale agreement, and also accepting that you should never just change your medical care based on something that you see on this website. And of course, there's a nice place right here that you could sign up to get updates about the GenCC, like I mentioned previously. So when you accept that, you'll come to this main database view, and this actually has all of the classifications displayed on one page. You can easily type to filter by gene symbol. At this point, we can only accept the current gene symbol. Scott is going to work on the functionality that the ClinGen website has, that if you were to search by an old gene symbol, it would still route you to the correct gene. But as of now, that functionality isn't here, and that's mentioned in the FAQs too. You could also filter by the submitted disease. These are all type aheads, or by the submitter. You can click and unclick that. You can easily filter by all of the clinical validity calculation or classifications that we have here. And if you wanted some more information about them, we have some info buttons. And so this will take you pretty much to the Delphi survey information that I talked about. And actually, which is kind of helpful, the harmonized definitions that we came up with for the Delphi survey, in case you would like to see those. So at this main page, there of course are different link outs, but I just want to show you that if you expand, so you'll see the number of disease equivalents. And so these are the map disease terms. So basically, if someone submitted on an OMIM term and someone submitted on a Mondo term and they're synonyms, that would be one disease equivalent. The equivalents are the monarch disease ontology term that it's mapped to. And then it tells you the number of submitters. And so you can actually expand this and just graphically see very quickly who the submitter is, what their disease is that they've chosen, and what their submission or what their clinical validity is, just right here. And it's very helpful for the ones that have multiple disease assertions, and they're grouped by submitter. And it shows you every single disease. So if multiple, in this case for ClinGen, if multiple classifications are classified as a particular clinical validity, they'll all just get listed together on the same line and just spill over to the next line. To show you some of these overall top ones before I get into any of these gene-specific ones, I should mention you can just click and unclick to filter these. If you were to go to the FAQs, there's some very nice FAQs there. Why is the GenCC important? What are the goals? What's gene disease validity curation? Can I submit to the database? So here is what I was talking about, Deb. Can my group join the GenCC? So most of the time when we ask groups, or when groups ask us if they'd like to join the GenCC, we basically point to they need to have a systematic way that they curate gene disease relationships that's granular enough that they can point to their assertion criteria either on their website or in some sort of document. And if it's a smaller institution, we really would mostly require that they curate up to ClinGen standards or use the ClinGen framework.
Okay, I see. Okay, so for cancer groups, I think there would be a lot of work needed prior to then getting it in this format in the kind of ClinGen standards, not for hereditary cancer gene curation that we do in ClinGen, but for external cancer groups. Okay, great, good to know.
Mm-hmm. Yeah, and not to say that every single group needs to curate using the ClinGen framework. For example, some of the clinical labs have developed slightly altered versions, but they are relatively granular, and they do use the clinical validity terms that we all harmonize to. And then there's just some other FAQs, and then there's some really nice screenshots of all the different pages that explain the different pieces of the different pages if you'd like to take a look at this at some point. There's also a statistics page. So this takes you to the statistics that I had shown you previously here. And then there are also statistics for each submitter. We're working on, based on multiple suggestions, getting a total for every single person right now, because you pretty much have to add it on the fly yourself here. But we'll hopefully release that sometime soon. And then you could easily click on a submitter page, and the personnel will be listed, so who you should contact if you have a question about a curation that is listed from this submitter, and then their assertion criteria, a little description about them, and then their visualization, the classifications visualized, so how many definitives and so on, and then all of their classifications here if you'd like to see their particular classification. So if you were to go to a particular gene, you can click any different number of ways. You can click the details here, you can just click the gene, and these will take you to a little bit more information besides just the drop down to expand. So we have 3 different ways to view the data. You have a little bit of information about the gene up here, and we're actually going to try and harmonize and make this look a little bit more like the gene banner and the ClinGen website, where there's just a few more link outs to learn a little bit more about the gene. But there are 3 different ways that we sort these things. So the first one is by classification, and we have the highest classification listed first. And so you'll see all the classifications here with their corresponding colors that correspond to those on the previous page, the gene and the HGNC symbol, the disease, and the monarch term disease equivalent that it was mapped to. So in this case, you can see PanelApp Australia actually did submit with an OMIM term, but it got mapped to a monarch disease ontology term. And so it shows which one it was mapped to, but which one it was also submitted. And there are link outs to both if you wanted to see the mode of inheritance. And if you mouse over it, it will tell you the whole word for that. And then the submitted date. So that's this, and this really was when it was submitted to the database, so probably not as important to you if you're doing a curation. You really want to look at the evaluated date. So this is when they looked at the gene-disease relationship. And as I mentioned, it really could differ based on when the curation was evaluated. So in this case, this one was in 2020, this one was in 2018, much newer. And then the group who has done it, and if you're interested in a particular classification by a particular group, you want to get some more information. I think this is most helpful for ClinGen curators. You click on it and you get a little bit more information. So in this case, there are Pubmed IDs here. Sometimes there's a public report. So here's a public report of the PanelApp panel for this gene. And sometimes there's even a little summary paragraph, much like there are for ClinGen gene disease relationships. So I would suggest if you're doing a curation for a ClinGen GSEP that first check the ClinGen website, because I mentioned it's not immediate if something's published on the ClinGen website that it comes over here. So I would search the ClinGen website, make sure it's not, and the gene tracker, and make sure it hasn't been curated by another group that you would want to reach out to and try to collaborate with, another GSEP, I mean. And if it's absent from there and the gene tracker, you could then come over to the GenCC and just go back to the, just go to the gene level page. And I found it most helpful to just search by gene symbol and see if it's classified in here. And then if you do that, you can click into a particular submitter's entry and find the Pubmed IDs that they're using for this assertion. I know that my participation in the syndromic group, it's also been quite helpful that for some of the biochemical function and physical interactions for experimental evidence for gene curations to see if a gene necessarily hasn't been curated by ClinGen that's supposed to be interacting with your gene of interest, and you're fairly certain that it's a definitive gene in the field, you could come and search it in this database and see what other GenCC submitters are saying about that gene disease relationship, and potentially bring that back to your GSEP to decide whether you want to curate that other gene to use as a piece of evidence or take their classification if they're using the ClinGen framework. So that is another thing that you could potentially do. I'll go back to the gene specific page. So the first one is by classification. Then you can see by disease, because maybe that's something to pay attention to. Maybe the disease that you're curating on isn't necessarily any of these diseases or a synonym to any of these diseases that are represented. So you wouldn't want to just immediately go back and take the Pubmed IDs that were listed here and just stick them into the curation if the disease isn't necessarily consistent. So be sure to pay attention to that. And of course, there are link outs if you want to read a little bit more about their particular disease that they've submitted. And the last one is just by submitters, which is kind of mirroring what's on the main page, but a little bit more information with all of the further details about the classification. And then each one of these, which is probably not necessary for a record that only has 3 entries, but you can easily filter out in case it's a classification that has like 10 entries or something, and only look at the classifications that are disputed if you really wanted to see what was the problem with a particular disease. Or you could filter by disease, you could filter by mode of inheritance, or filter by submitter within this. And then if you're interested in just looking at the data or using it for separate work, for example, not any global analyses, hopefully until we publish our paper, but there's also a data download in a few different formats to be able to download the data and just look at it in an Excel spreadsheet. And then if you ever had any questions or concerns, of course, you could always reach out to me, but there's a contact button that you could contact just the general GenCC at thegencc.org, which, as I mentioned, comes to me, Scott, and Heidi. And I think that is all as far as the database. So as I mentioned, I just search for your gene of interest and then kind of just click around to get all the summary information that would be helpful for you and your curation, particularly the Pubmed IDs or maybe a comment about the gene disease relationship from a particular submitter. And if it helps you, also you might want to go to the submitter level page and read about their assertion criteria briefly to help to understand if the way that they're classifying genes is consistent with how we're thinking about it within ClinGen. And so I think that is all I have, Jenny, so I can take any questions from anyone, or if anyone else would like to see any other functions that they don't think I touched upon, feel free to ask questions now.
Well, this is great, Marina. Just a huge amount of work, and it looks fantastic, and just, yeah, so incredibly helpful. I mean, I really like that use case you gave for example, an interacting gene. I think that's you know, certainly somewhere it could be really helpful, and just finding other cited literature from other groups that have curated the gene. Certainly, I know that the information that other groups have shared with us, like Council now, Myriad Women's Health, for their curations, you know, just having the PMIDs and that previously curated information has been really helpful. So yeah, this is really great. Does anyone have questions for Marina or anything else you'd like to see?
Marina, this is Deb. So I can see on the website that that there are assertion criteria for it, but sometimes those assertion criteria can be really different. Like you talked about, like with the cancer, you know, ones is very different from currently the ClinGen way. Is there a score or some understanding of the differences of the assertion criteria? Like some of them are more different than others if they're not using exactly ClinGen gene disease validity?
There isn't necessarily a score right now. We are hoping to talk about that a little bit in that flagship publication. I would say because we're on a ClinGen group right now, let me go to the statistics page. I would say that many of the clinical labs are very similar, if not using exact ClinGen framework for their classifications. OMIM is OMIM, as you know, so it's really just like published literature is there, and there's a gene disease assertion. Here it is. And that's why we have the term supportive for OMIM, even though their pilot's not ready yet. Orphanet's kind of the same way, where it's basically there's a gene disease relationship. Here it is. At least from what I understand, talking to the team based in France, this is for really rare diseases, and they often have to have something present in Orphanet to be able to have the government pay for testing for it. So these ones are much, much less granular than, say, some of the ones that are either the ClinGen framework or very similar to the ClinGen framework. The PanelApps are interesting. As I mentioned, these websites basically have a bunch of gene panels, and they're evaluating whether a gene should go on that panel for a particular feature. So it's a little bit of a different way to think about lumping and splitting. It's much more panel design or indication-based analysis in that case. So those ones I definitely think are a bit different too from what we do. And
yeah, that's kind of how I was wondering. It, like the ones, so you mentioned like by the ones that are similar to ClinGen are most of like the clinical labs, and then you know the one I clicked on was Genomics England PanelApp to look at that. So yeah, any way to kind of differentiate those, because you would like if there's a discrepancy between the ones that are using the ClinGen approach, that's an issue, right? But if there's a discrepancy in between the other ones, that could be a discrepancy between their curation approaches, right?
Yes,
okay.
It definitely could. And I think that we'll need to find a way to frame it, because we don't want to make the database too ClinGen centric, because everyone is kind of an equal partner of GenCC. So at least as of right now, we've just tried to make it transparent and say these are everyone's assertion criteria, because just like in ClinVar, I mean, to be fair, most people are using ACMG guidelines, so maybe it's not a good example. But just like in ClinVar with, I don't know, an AI literature mining assertion, that's clearly going to be very different from a clinical lab's assertion. And really the only way you can tell is by their assertion criteria, at least at this point. So we'll try to make it maybe a little bit more transparent in the publication and maybe somewhere on the website. But we definitely need to think about that when we're looking at discrepancies. And that is another difficulty, I think, with discrepancy resolution. And one of the GenCC submitters pointed out that conflict resolution isn't necessarily a good word to use, because it really should just be, we're pointing out differences, particularly if the way that you classify gene disease relationships are different in the first place. How much can you compare it? So you're right, that is a good point, Deb.
Yeah, thanks, Marina. So there is another question in the chat excuse me, how are the PubMed IDs selected? For example, for one of my genes of interest, the IDs listed only describe the clinical phenotype, not any genetic data. So I would assume, does that just, excuse me, depend on the submitter and the type of information that they are citing? I guess
I really think it is submitter specific at this point. And you might want to look at their assertion criteria, but that's also why we've listed a representative for each group, that if you would like a little bit more information as to how they cite their PubMed IDs, you could always reach out to them. And they all know that their information is public. But yeah, this really is like a ClinVar for genes. So feel free to contact me with general questions, but if you really do have questions about how people are classifying things, it might be most helpful to reach out to that particular submitter, because these really are a database of assertions.
Any other questions? Yeah, and I mean, I think moving forward, it is going to be really interesting to look at how these differences are resolved. I mean, how many of them can be resolved just by the date that the information was curated or the framework versus, you know, any just differences in how people are interpreting some of the data. That's going to be interesting to see moving forward.
Yeah, we've already started to have discussions too about how classifications change over time, and potentially adding another field, just like we were thinking about in ClinGen and the gene curation interface, for collecting the first date of publication of human evidence, just so that people can see that too, that maybe a limited gene that was just published last year is a little bit different than a limited gene that was published 20 years ago, and really